ladies and gentlemen, we are going to get started into the cell cycle unit, and what better way to start off the cell cycle unit than talking a little bit about mitosis. That's right, the process of cell division. So we're going to take a look today at how cells, one cell, can become two cells. How does that process take place? What are some of the mechanisms in which that cell can break apart into two independent cells and complete the process over and over again, making an organism that is trillions and trillions of cells big. So, your objectives today, by the end of this lecture, you'll be able to explain the process of the cell cycle and the importance of each step in the cell cycle. So, what is prophase, metaphase, anaphase, telophase? What goes on in each step that helps the cell cycle take place? Additionally, you should also be able to predict the end products of a cell going through mitosis. What do we have at the start? It's really important to know what happens at the end, because we're going to talk about something else called meiosis. And it's important to know the difference between what happens at the end of, or what you get at the end of mitosis versus what you get at the end of meiosis. So the first thing we need to understand is that cells do have to divide. It's just the law of nature that cell size is limited, so as the cell continues to develop, it gets larger. It's harder just to maintain homeostasis inside of a larger cell compared to that of a smaller cell. So making copies of the cell is important. We all know that cell size is limited, so the process of cell division is really important for cells to be able to maintain that homeostasis and to be able to maintain the balance inside the cell. So in order to understand this, we have to understand that DNA gets condensed in the process of mitosis, and they make what are called chromosomes. Chromosomes are just condensed DNA, and that condensed DNA is also known as chromatin. So chromosomes are made up of chromatin. Now, we have to understand that in order for the cell to divide, DNA has to replicate. So after replication, the cell looks like an X. It looks like the picture that you have on the right. Those are two chromatids. Each chromatid has identical genetic information. So there are two of those chromatids there with identical genetic information. Each half of the chromosome is also known as a sister chromatid because they're genetically identical, and they're held together in the center by a centromere. So you have a chromatid. It undergoes the process of replication to become a chromosome. Those chromosomes are then going to be pulled apart so that each new cell is going to have the same genetic information as that of the original cell. So that's why DNA has to replicate, is so that each cell after mitosis has the same genetic information as that of the original cell. So keep in mind, chromosomes are a very important part of mitosis. That we have an X, which is what it looks like after replication. Prior to that, it looks just like a stick. All right, and after replication, it looks like an X, and then they're pulled apart during mitosis. The cell cycle, and again, it is a cycle, so this process will continue over and over again, involves primarily just growing and dividing, and there are three main stages to the cell cycle. The first is known as interphase. This is the longest phase of the cell cycle and primarily involves just simple growth and normal function of the cell. The cell is just doing what it normally does. The next stage is what is known as mitosis, and that's the division of the nucleus and the DNA. And then cytokinesis, which is the last step, is the physical division of the cell. So we have several steps here involving the cell cycle, including interphase, which is normal cell function, mitosis, which is the division of nucleus and DNA, and cytokinesis, which is the actual physical division of the cell from one into two. Interphase itself, which again is the longest phase of the cell cycle, is divided into three phases: G1, which is normal cell growth; S phase, where DNA replication occurs. S stands for synthesis. So in S phase, we're going to be developing our chromosomes and making copies of them so that we get that X shape. The last part is G2, which involves a lot of chemical producing in order to prepare the cell for cell division. So again, interphase divided into three parts: G1, S, and G2. Mitosis itself is divided into four phases: prophase, metaphase, anaphase, and telophase. I always remember PMAT as the best way to remember this. Just a quick little abbreviation for prophase, metaphase, anaphase, and telophase. During the process of prophase, the chromosomes condense, so we get those chromosomes that we've been talking about, and something that's called spindle fibers form from the centrioles. The centrioles are the organelles that aid in cell division. You may remember that from our cell unit. These spindle fibers are going to attach to the chromosomes. These spindle fibers allow for the chromosomes to be able to move inside of the cell, and eventually we'll be pulling those chromosomes apart, giving each side of the cell identical genetic information. The nucleus also begins to disappear, which allows those chromosomes to escape and roam freely in the cell. Once they get attached to spindle fibers, they move on to metaphase. Metaphase is the phase of the cell cycle where chromosomes line up in the center, along what's called the metaphase plate. So you see in the picture to the right, all of the chromosomes that are within the cell are lined up along the metaphase plate and attached to spindle fibers. These spindle fibers help put the chromosomes into place into the center. This is very important because if they're not in the center, the spindle fibers may not pull the chromosomes apart correctly, resulting in an abnormal amount of chromosomes in one cell compared to the other. So again, metaphase chromosomes are lining up in the center of the cell. Anaphase is where the spindle fibers begin to contract and the chromatids are pulled apart by the spindle apparatus. This ensures identical genetic information for each half of the cell. Those chromatids are then moved towards the poles of the cell, closer to the centrioles. So they're being pulled apart from the center of the cell in anaphase. In telophase, the chromosomes decondense. The chromosomes start to not be so tightly coiled anymore. The nucleus begins to reform, and the spindle apparatus disappears. What we're seeing here is the beginning of the creation of two new cells, as we start to see the cytoplasm begin to divide. Eventually, cytokinesis occurs, where that furrow in the cytoplasm pinches inward even further, and the cytoplasm physically splits one cell into two. So what we end up with is two cells with the same genetic information, because those genetic chromosomes were pulled apart during anaphase. Well, like I said, what we end up having here is two cells that are genetically identical through the process. After cytokinesis, interphase starts again, and we move on to G1. What's important to understand here is that this is a cycle. So again, at the end of cytokinesis, we start over again. The whole process continues over and over again. Interesting fact is that the average cell can divide about 50 times before it dies. So it makes you think about all the different cells that are inside of your body and how many times they can multiply and how many new cells are being produced over a period of time. Key concepts to remember though is at the end of mitosis we have two cells with the same number of chromosomes as the parent. So human body cells have 46 chromosomes, therefore at the end of mitosis each cell should have 46 chromosomes. They are genetically identical to each other. So every skin cell that you have should be genetically identical to the one next to it. All of those cells are continually performing mitosis and hopefully making cells that are similar to that are around it. Additionally, all body cells perform the process of mitosis. All body cells perform this process, with the exception of sex cells. Sex cells are produced through a process called meiosis, which we will talk about next time. But every cell in the body, except for sperm and egg, are produced by the process of mitosis. So hopefully, by the end of today, you'll be able to explain the process of the cell cycle and the importance of each step in that cell cycle, including interphase, including G1, S, and G2, mitosis, which includes prophase, metaphase, anaphase, and telophase, and cytokinesis, and explain a little bit about what happens at each step. Additionally, you should be able to predict the end products of a cell going through mitosis. You should know that they had produced two genetically identical cells with the same number of chromosomes as before, and that all cells in the body perform mitosis, except for when we're producing sperm and egg. If you have any questions, be sure to email me to get some clarification, and make sure to take your video quiz at the end of this lecture. Have a great day, guys. We'll talk to you later.